Okay, Matt.
Okay,
ready to
thanks.
go? Right.
Yeah, so as Neil said, I'll be talking about some challenges in the automated spatial analysis of 3D segmentation and cell typing. So my group develops standardized computational pipelines that are run on all data sets of a certain type, given for both HubMap and SendNet, but this work is primarily HubMap focused, hence the regalia for the slides. Yeah, so I'm presenting on behalf of especially my imaging subgroup, and I'll get into some of the people there. So I'll be presenting two different tools that we have developed recently. The 3D cell composer tool, which is a 3D cell segmentation method that leverages 2D cell segmentation. And then RIBCA, a robust image-based cell annotator. So that's a cell annotation method, completely automated, that is robust to pixel noise, et cetera. And we'll get into some details about both of these methods. So first, we'll go into 3D cell composer. So for 3D cell segmentation, even when you have a nicely registered, aligned, reconstructed 3D volume, there are really significant challenges in producing that 3D segmentation mask. For cell and nucleus segmentation, then of course getting the boundaries of both of those, if you're looking at boundary protein expression. A lot of those challenges are from limited training data availability. More and more keeps coming online, but still expertly labeled 3D cells are not as easy to find as expert 2D cell segmentations. And there is inherent computational complexity in the additional spatial dimension there. And some of the existing 3D segmentation methods, even if they work well enough for one data type, they often don't generalize that well to new imaging modalities, even if you're using a similar type of channel like cell or nucleus marker. Okay, so 3D cell composer leverages high quality 2D segmentation methods for any applicable data types. So especially what we have represented in HubMap in terms of public data sets, that's imaging mass cytometry. There aren't many of those data sets, but they're pretty rich, and it's a nice Z dimension or Z spacing compared to the X and Y pixel coordinates. And multiplex immunofluorescence, Codex PhenoCycler and CellDive. So those 2D segmentation methods we have, those would extend to 3D pretty nicely, but this method is applicable in principle to any 3D data that for which you would reasonably expect to see the same cell in multiple Z slices. So if your Z spacing is less than about 10 microns, 5 would be great. Any more and finer resolution than that is even better. And anything for which you have a well-performing 2D segmentation method. So there are many that are validated, papers come out all the time with new segmentation methods for different marker membrane stains, et cetera. A lot of these work really well for different 2D data types. And this allows generalizing these to any 3D data that you might get of the same assay. Okay, so most generally, we run 2D segmentation on every slice. So the X, Y slices in the Z dimension are straightforward enough. We reconstruct that 3D volume, run also in the XZ and YZ dimensions. So there's some extra computational costs there of performing those multiple segmentations. And then we get an initial preliminary 3D cell on each axis just by stacking those. And then some algorithmic approaches to repair the 3D segmentation to find out that, yeah, this voxel assignment is actually the same across these different slices. So I won't go into the details of that algorithm. That's I'll pull you to the preprint for that. But some of the segmentation results, this is for one of the 3D IMC data sets in the HubMap data portal. So the output is the voxel mask, same as you would get as a 2D segmentation method, indexed OME TIFF with different channels for cell, nucleus, and those boundaries. Conversion to GLB format, so you can load this in any standard 3D tool. There was a screenshot of Blender that I happened to pull up in the last day or so. Some of these mesh files are actually quite large, though. So there are significant challenges in visualizing these. That's why I appreciate the other presentations about the optimizations and how this can actually be viewed. Not only the representation, but also the rendering. So that was really nice to see. Okay. Yeah, and then QA and QC measures in JSON format. So that comes from our group's unsupervised SPRM tool, which I'll get into some details in a moment. So comparison of some results from 3D cell composer with different 2D segmentation methods wrapped for the XY, YZ and XZ dimensions. So deep cell versus cell profiler in the top two panes. Then native 3D segmentation methods, the cell pose 2D to 3D and 3D cell seg not performing nearly as well on the 3D IMC data sets that we were experimenting with. Okay, then I mentioned the QA/QC measures. So the cell segmentation evaluator package is integrated into HubMap data processing. You get these outputs for any standard HubMap data processing result. So this defines metrics defining a good segmentation without a human reference or human segmentation as reference to say that this is where we think the cells are. It's really computing measures to say, this is what we think a good segmentation result would look like. So, uniformity of expression and things that you define, or that you note as cells. How good is this image in terms of, a quick foreground background calculation? How much of those foreground pixels or voxels are occupied by cells? Variation in cell size. Yeah, a lot of these other measures were defined for the 2D case. Some of them generalized naturally to 3D. Some of them actually dropped due to, shape calculations not being nearly as straightforward in 3D. Okay. But then across all of these measures, each one of these gives you a metric for that image. And looking across all available image, we then fit a PCA model to explain the variance in these metrics. And that first principal component, since these are all defined as a higher score being better, can give you an overall quality of the image and the segmentation result. So higher in this PC1 is a better segmentation result with a higher quality image. And that we're assigning to a deep cell with the membrane marker. Deep cell cytoplasm marker is here. And the first symbol here and then the additional points afterward are with adding Gaussian noise. So how does this quality drop as the image gets noisier and noisier? But the overall takeaway message here is that the deep cell membrane and cytoplasm markers do very well and aren't very robust to noise. So we see some drop in principal component 2, which captures some less important the features that we computed. Cell X performs reasonably well in PC1, but when you start adding noise, the performance drops off pretty dramatically quickly. All right, so yeah, takeaway message is that deep cell with membrane and cytoplasm run through 3D Cell Composer outperform the native 3D methods, 3D Cell Seg and the Cell Pose 2D to 3D. Okay. And then, yeah, I mentioned the unsupervised analysis via our SPRM package. So good, that does display properly. So this computes a lot of unsupervised features from each cell, each nucleus from the image data, performs multiple cluster methods. So clustering via all available features with a UMAP embedding is what's shown here. So my group does not have a ton of expertise in 3D data visualization. So this is just a quick animated PNG going through the Z slices up and down and then back. So this, I think, is 22 coordinates in the Z dimension. It's about 580 in the X and Y for this IMC data set. Note that we actually have cells on the border, the X and Y and the Z dimension. But if we don't have an entire cell, then that's dropped from the unsupervised analysis. It isn't really meaningful to cluster a cell based on what might only be 40% of its protein, or no, this is IMC, 40% of its expression. Okay. All right, a few other ways of clustering cells. These might be more or less meaningful. So clustering cells via mean protein expression. So that's the expression vector divided by the number of pixels, or sorry, here it's voxels. Clustering of nuclei only via total protein expression. And these all give you different views of what's happening in different spatial parts of this tissue and how different cells group together based on, let's say, shape, if you have that, versus mean or total protein expression. Okay. And yeah, all these results are available for any HubMap process data set, so you can get these on the HubMap data portal. A lot of these, the same cluster assignments are integrated into the test view, but we still have the PNGs or APNGs that you can get from the dataset directory. All right, so yeah, 3D data processing is a hugely complicated topic. This is just one slice of what we're doing with the 3D segmentation and then the unsupervised analysis afterward. So I'll go in a different direction after this with the cell type assignment tool that we recently released. So we're calling this the Robust Image-Based Cell Annotator. Okay. And so we'll note that a lot of current methods for assigning cell types rely on your average marker intensities. Some don't use spatial information. Some don't use subcellular protein localization inside the cell. And a lot of these require tools for annotating with specific marker panels and are really sensitive to which markers are in which panels. So a possible need to retrain models, for example, if you have a slightly different marker set for this experiment versus the others, or I think even different markers from different Z slices in the same experiment, as happens often enough. And yeah, a lot of these image-based models can still have that sensitivity. So that motivated work in this image-based cell annotator. So it crops the tissue image after cell segmentation, gets the single cell region for each with some soft masking, and applies multiple vision transformer models from different panels to each to give a cell type assignment, which is an ensemble of cell type assignments from the different models that they're actually voting on, then with confidence scores. Okay. So here we have the 5 different trained models that actually are with the package. As we'll note later, that this is extensible. You can add different models due to the structure of this, in addition to iterative improvement of these models if you have lots of data sets with the same markers. So some of these are more or less specific. Kind of the base immune panel gives you, 1 vote with more specificity and more confidence in cell types from some of the more robust models in terms of the number of markers that are present. Yeah, and each of these is a vision transformer. So it takes this soft masked cell image, breaks up into patches, actually iteratively trains on subsets of the package to learn not only the relationship between the pixel data and the cell type assignment, but relationships between different portions of the image. So being robust to noise in each image, the pixel data for each cell. And this all feeds into multiple vision transformers to give votes for which cell type that cell is. Okay. Yeah, noted that each of those, and all of these models vote, and we'll actually show the confidence scores coming out of that in a moment. Okay, so for the model training that we were able to do, so this was lots of things across the human protein atlas images containing more than 10 million single cells, consolidated all the labels to 17 different cell types from the curation of the cell labels that we had. But a huge issue that came up in the training also was heterogeneity in the marker panel. Not only just like spelling a certain protein differently, but when one is present and one isn't. So RIPCA incorporates an imputation method to replace a limited number of missing markers. You don't want to go more than 1 or 2 or 3, inferring marker expression from what's there, but it actually works quite well. Okay. And so with a few of these panels, the immune base, extended, and then full. So the prediction performance, if you're leaving the channels blank, that's not a great approach. You can get performance much closer to your baseline accuracy, even if you're removing 1 or 2, or for the panels that have more markers, 1, 2, or even 3 missing markers. You can push this up close to your unperturbed accuracy. It's much better than dropping those channels or assigning a missing value or value like 0. So RIPCA is robust to not only heterogeneity in the panels, but noise in some of the images. Yeah, and that increase is quite significant. And so we applied this to the HPA tissue microarray images, distinct from the training data, and HubMap codex tissue images in bulk. And this produces not only cell type annotations, but confidence scores. So these are cell type assignments from the HPA tissue microarray images. So different tissues showing different localization of B cells, CD4, CD8 T cells, different anatomical structures. Okay. So with validation, very high fidelity recreation of expert annotations. So lock everything on the main diagonal being quite large values. Some confusion here between what is that? Smooth muscle and epithelial cell. All right, and so these are the HPA results, the cell types across different tissues. So some heterogeneity in performance between tissues, especially liver, ovary not having a large proportion of cells marked other. A lot of this is due to the panels that can be applied to each of these data sets. If the markers just don't match, we can't impute more than 1, 2, or 3. There isn't much to do besides call that cell as other. Okay. So we have more detailed results of a breakdown for the HubMap codex data. So applied this to 88 and counting. So there are, I think, more in the HubMap data portal that we're extending the RIPCA results continuously. And there's, as we said, a lot of heterogeneity in the markers, even within data sets from the same tissue provided by the same group. So things that are run on different days don't often have the same markers. And some of these, the models and panels aren't usable for those tissues due to too many missing markers. And as we said, if a confidence score is below a per cell threshold, you get the unknown cell type. And this actually shows the cell type assignment results across all of the codex data sets in HubMap that we were able to run. So these are all public data sets, and these are the cell type assignment results from this method. So, and also noted as which panels were applicable to that data set based on matching the markers. So the more panels that were able to be applied, generally the better results. Some of these data sets that had only the basic immune panel applied, of course, you're going to get a large proportion of other cell types assigned. And yeah, as we said, heterogeneity even between the panels that are applicable inside, for example, lymph node and spleen and thymus. Typically a lot better coverage for large and small intestine. So thanks to the Stanford TMC for that. And for one particular data set, this is an image colored by the cell type assignment on the left, and then the confidence score given by Rybka on the right. So not only can you say that this is a B cell, but this is confidence 0.7 versus 0.5. Set a threshold there for your own analysis if you would like. All right, and then with these cell type assignments, it's worth going into how exactly do we show these in the HubMap data portal? How are these used? So I think thanks to some very recent work from Nils group, these cell type assignments can show up alongside the unsupervised clustering assignments. But our method SPRM that we mentioned is the unsupervised analysis tool. That's now an entry point also for cell type assignments. So anything that comes out of any possible tool that we are able to use. So that includes Rybka, deep cell types from David von Valen's group, Stellar, as Yuki mentioned earlier. So in addition to passing through those cell type assignments, we perform subclustering of each cell type annotation. That'll give you, this B cell, this set of B cells clustered into a few different groups, and you can view those spatially to see is there a difference in location. And that also includes the other unknown cells. So if we don't know what it is, then you can say it's still cluster 1, cluster 2, look at the markers between those, and that can be quite meaningful. Right, yeah, and actually things will show up alongside the cell sets there in Vitesse. Thank you, Nils. So this is a cell type assignment plot here. So B cells in green, cytotoxic T cells in red, and so on. And each of these panels here is a specific cell type from panel A subclustered to say, do these cells group together just based on protein expression or based on the available features? So among the B cells, there's a red cluster here, which localizes in that anatomical structure. There's a ring of blue cells around and green here. So that's from whatever available cell type annotations. Is there enough signal to be able to pull these apart even further and then look spatially to see, does that make any sense? And clustering the other cells can be free for all, but a lot of these specific cell types still do so with that spatial localization. All right, so there's also a Napari plugin. You can interactively run and analyze these cell type assignments. What we're working on currently with Rybka is 3D cell subtyping, 3D cell typing based on some of the similar techniques that we showed in 3D cell composers. So, apply to different slices of the image. There's some additional computational cost there. Now a collection of additional training data. So as I mentioned, this is a modular design. You can iteratively train each existing model with additional data that you have. And you can add additional panels, additional models, which then get to vote the same as the 5 that are mentioned in the results here. So deployment in the HubMap data portal, so that's actually complete, along with deep cell types. And I think the automated runs of those are, I think, in QA status on some part of the HubMap portal. So you'll be able to look for automated cell type annotation in HubMap extremely soon. All right, I'd like to acknowledge funding from the NIH for HubMap and for SendNet. My group members and other investigators, especially Bob, Ted, and Haran. Haran was a PhD student who since moved on for a lot of this work. This is my imaging subgroup of that HubMap computational tools component. Lots of collaborators in HubMap, especially thanks to Nils group and the Stanford TMC, and everyone at VUES for hosting. Thank you.
Thank you for this talk, Matt, and happy to take some questions if there are any in the audience.